So she's been instrumental in moving that project forward. And she's working with John and I on our reading studies, reading and brain studies. So she'll take it from here? Sure. Okay.
All right.
Will we get these slides?
Yeah, I already put them in the Dropbox.
Oh.
Yeah.
So I'll follow along. So while everyone's looking that up, you guys have had the benefit of getting to know John and Joanna over the last couple of days. So I'm just going to give you a little bit of background of me and who I am and what I'm bringing to the table. As Joanna mentioned, I'm a postdoc in a couple different places. I got my doctorate in the spring of 2013 in neuroscience from the University of Texas at Dallas. So my expertise, my training really is auditory neuroscience and genetics of language disorders. So how does the brain process sound, specifically speech sounds? And in kids with dyslexia and language impairment, how are their brains processing sound differently? And what are the genetics of those differences? Again, we're going to cover kind of a lot over the next hour. A lot of it's going to be probably new to some people, so feel free to stop me at any point if you have questions or things that you'd like me to talk a little bit more about. So this is kind of our general agenda for today. We're going to talk about some common misconceptions of genetics. We'll go through an abridged genetics primer just to give you guys some basic vocabulary and knowledge about genetics. And then we'll talk about the genetics of human disorders, candidate genes for reading disability, imaging and genetics, and then finally some implications of genetics. And by the end of the next hour, you will hopefully be able to answer the following questions. What does heritability mean? How are dyslexia and heritability related? Are genes deterministic? How can epigenetics cause human disorders? And how can a gene influence behavior? So genetics is kind of a hot topic right now in the media. I did a quick search for headlines on genetics and had to whittle down hundreds of headlines saying the gene for IQ, the gene for obesity, the gene for religion, is a gene for everything, right? But in most cases, there's not one gene responsible for these things. The media tends to simplify the story to make it accessible for everybody, but there is not one gene responsible for religion or IQ or obesity or any of these things that we talk about. There are a variety of genes. There are some disorders where a single gene can disrupt IQ. For example, Fragile X is a single gene disorder that causes impaired IQ. But in most cases, it's multiple genes contributing to a disorder. Even if there's only one gene, chances are we still don't know how it contributes to the disorder. And even if we do know the mechanism, it's rarely the only thing. So it's really a more complicated story than the headlines in the media will have you believe. So just take those headlines with a grain of salt is kind of the takeaway of this whole point. Of course, there are instances where one gene is involved and influential. We've all probably seen these headlines. We remember the Angelina Jolie story of her preventative mastectomy. BRCA1 and BRCA2 are two genes that are highly predictive of breast cancer risk. If you have a mutation in one of these two genes, it's highly correlated with your risk for breast cancer. However, again, most human traits and disorders are controlled by tens to hundreds of genes. It's usually not a one gene thing. Language, reading, and learning are examples of these complex genetic traits. Another misconception out there is that if something is caused by genetics, it can't be changed. Certainly there are things caused by genetics like eye color and height that cannot be changed, but there are plenty of other genetic conditions or traits that can be changed. For example, autism is extremely heritable. It is caused by many genes. I did a quick Google search this morning, and the last count, there's anywhere between 400 and 1,000 genes that are involved in autism. And then we wonder why we don't have a cure for autism yet. It's a very complicated story. But autism can be treated. There is a very effective one-on-one enriched environment therapy that can raise IQ and improve communication skills in these kids. So even though this is a genetic disorder, there are ways to mediate the risks and the symptoms. There are also plenty of experiments in rodent models. In my doctoral lab, if you knock out a gene in a rat, you can fix a lot of the neural problems via behavioral training. We'll talk about that a little bit later as well. So let's talk about genetics. There are 23 chromosomes in the human body. They're located in the nucleus of every cell in your body. You have 2 copies of each chromosome. Some of this should be familiar from biology in high school. Your egg and sperm cells are the only cells in your body that only have one copy of each chromosome, and that is to be passed down to the next generation of progeny. Each chromosome is composed of a single folded up strand of DNA, or deoxyribonucleic acid. And these 23 chromosomes contain your entire genome. So every cell in your body contains every gene you have. It doesn't mean that every gene is expressed in every cell, it just means that every cell has the code for every gene. So a gene is a segment of DNA that codes a specific protein. And because DNA is one long continuous strand, there are markers for the start and stop of every gene, and they're known as codons. There are 20,000 genes in the human genome, which seems like a lot, but we do not have the largest genome, and I realize it's hard to see from the back. We are the second on that graph. Grapes have more genes than we do. So we like to think of ourselves as the most complex.
We didn't hear what you said, has more?
Grapes.
Great.
Grapes have 30,000+ genes in their genomes. So we may have the most complex brains on the planet. We do not have the most complex genome. So those 20,000 genes, they make up 2% of our DNA sequence. So that leaves 98%. The majority of your DNA sequence is not genes. Anybody have any idea what the rest of it is? Stuff. It is stuff. And there's a term for that stuff. Has anyone ever heard of junk DNA? Okay. I put this in quotes because it's not really junk. 98% of your DNA could not be junk. Nature and evolution would not allow that. But it's been known as junk DNA for a very long time because it's the part of your DNA that doesn't encode protein. So before we had techniques to really get at what those pieces of code were doing, people assumed they were just useless. And they actually have a lot of uses. They're used for a variety of things, including sensing chemicals in the cell, turning other genes on and off, they're regulatory factors, and evolutionary purposes. There are a lot of pieces of code that do very similar things. And so when you think about evolution passing genetics down to the next generation, the redundancy allows for some natural selection. Which pieces of code are going to do the best job and which ones can be mutated, you'll try again, and hopefully you get a better piece of code in the next generation. Human DNA, we are 96% the same as chimpanzees. We know we are genetically very similar to those primates. And we're 99% the same across other humans. So in this room, our genomes are 99% the same. So that leaves 1% for variation. That variation is known as alleles. There are alternate forms of a gene. So the DNA sequence is different across individuals. Not all of these variations cause disorders. Most of it's just what makes us unique, what makes my hair brown and curly and yours blonde and straight. So DNA, its structure is a double helix. I'm sure we all know this. It was discovered by Watson and Crick in 1953, which of course earned them the Nobel Prize. The nucleotide bases, the structure of DNA, is adenine, thymine, guanine, and cytosine. You don't need to remember those, but when you look at the diagrams of DNA and you see A, T, G, and C, those are your bases, and that's what all DNA is coded in. A single change is known as a SNP, or a single nucleotide polymorphism. We all have SNPs in our genome. There are over 3 million base pairs in your genome, and there's at least one SNP for every 300 base pairs. So everyone in this room has more than 10 million SNPs in their genome. So things that are switched around, bases that are switched.
Are these like mistakes?
It's mistakes, yeah, for the most part. Some of them are inherited. So you'll get some SNPs from your parents. Some of them are just random mutations that happen, most of which are harmless. Most of them are switches that don't really have any functional consequence. Your body's pretty good at understanding most of the time what the genes mean. But some of them are, of course, harmless. Most of them are normal variation, eye color, hair color, those kinds of things. And again, some of them are harmful. So sometimes a SNP can cause a disorder on its own, but sometimes you need multiple SNPs. for symptoms to appear. So I'm just going to talk briefly about the differences. So I mentioned Fragile X a little bit earlier. Fragile X is one of the disorders that is caused by a single gene and a SNP in that gene. So up here we have, this is a genetics family tree, so I'll just talk you through this really quickly. The squares represent males in the family, the circles are females, the black circles are affected individuals, and the white circles are unaffected individuals. So you can see in the case of Fragile X, if you have this one little mutation, you automatically have the disorder. One SNP means disorder.
Something we used to call like a dominant gene disorder.
Yeah. So Fragile X, for those that don't know, is the most common cause of inherited mental retardation. Most of these kids are comorbid for autism as well, so it's often lumped into the autism spectrum. And these kids have physical characteristics as well as mental characteristics. They have elongated faces, larger ears, so they're easy to kind of pick out in a crowd. Most language, cognitive, and behavioral disorders, though, are more complex. They're not caused by a single gene, and each contributing gene may have a small effect individually that then combine and interact with either other genes or your environment to cause the disorder. So environment can affect gene expression. So the structure holding DNA in place is referred to as the epigenome. This is another buzzword. Right now in the media, epigenetics is kind of the hot new thing. So we're going to talk about it, make sure everyone understands what epigenetics is. The epigenome adjusts its structure based on our interaction with the environment. So you have your double helix strand of DNA. It's wrapped around the epigenome, and the epigenome changes its shape based on the environment.
No, I'm not getting this.
I'm going to walk you through it.
Okay.
I know this is a complicated thing. We're going to go step by step.
Is it a thing?
It's a thing.
Epigenome, it's like
The
a...
epigenome is the physical, yeah, if you want to think about it that way. There's, I'm going to show a video in a little bit that's going to kind of break it down visually for you.
Okay.
But basically, it's the structure that holds DNA in place. Makes sense. And there are methyl tags, or methylation, that's on this epigenome. And that basically tells the epigenome whether it should be crunched up tight or stretched out and loose. And I'm going to tell you more about that. Okay? Bear with me. Okay. So when there's a methyl tag on your epigenome, it causes the epigenome to tighten and scrunch up. When the methyl tags come off, the epigenome relaxes and stretches out. And what that does is it controls which genes are readable and which genes are not. So think about the epigenome like, let's say a piece of paper. I don't have a scrap piece of paper, but let's say you have a piece of paper. Piece of paper is the genome. The words on the page are your DNA. If the epigenome is relaxed, the page is flat, you can read the words on the page. If the epigenome is tight, I crumple up a piece of paper and you can no longer read the words on the page. Does that make sense? Mm-hmm. So epigenome relaxed, you can read the genes. Epigenome scrunched up and tight, you can't read the genes. So this has a practical function in our body. Remember I said that every cell has every gene. But there may be some genes, for example, that are really important in your heart that have nothing to do with your stomach function. So in the heart, you want the epigenome around that gene to be loose and relaxed so that gene can be translated in the heart. Whereas in the stomach, the epigenome would be tight so that that gene cannot be translated where it's not supposed to be. Does that make sense? Yeah? So far? Okay. The methyl tags are added or taken away based on the cell or the organism's experience. That could be location in the body. There are chemical markers throughout your body that will kind of let the epigenome know where it is and what genes should be expressed and which genes should not be. But it can also be added or removed based on the stress in your environment and the things that you as an organism are experiencing in the world. These tags are passed down to your daughter cells, so offspring can inherit a parent's methyl groups. Sometimes that's really helpful. Again, different cell types need different methyl tags. But there is also an aspect of this that your experiences can be passed down. There are plenty of experiments in rodent models that show that stressed mothers raise anxious pups. So there is kind of that one-to-one relationship. And just again, to help make this make a little more sense, I'm going to show you a quick video. No, I have my sound? What's going on?
It's not personal.
I know, we tested it beforehand.
Sometimes so you just need to.
That's my car.
Okay. All right. Is this playing right now? Oh, okay. Let's play it. We'll get it to work.
I can hear it. Technical difficulties. Try again? There we go.
DNA contains the instructions for building all the parts of the body. But DNA is only half the story. The DNA in our bodies is wrapped around proteins called histones. Both the DNA and histones are covered with chemical tags. This second layer of structure is called the epigenome. The epigenome shapes the physical structure of the genome. It tightly wraps inactive genes, making them unreadable. It relaxes active genes, making them easily accessible. Different sets of genes are active in different cell types. The DNA code remains fixed for life, but the epigenome is flexible. Epigenetic tags react to signals from the outside world, such as diet and stress. The epigenome adjusts specific genes in our genomic landscape in response to our rapidly changing environment.
And the link to this video is in your slides. And actually this website has a lot of other interesting videos about the epigenome in general and ways it can be influenced. So I encourage you to take a look at that website.
So,
Yeah.
the histones are part of the epigenome?
The histones are the physical structure. So the DNA is wrapped around the actual histone,
Uh-huh.
and then those are connected.
And so it's, methylated histones are epigenome.
Yes, it's the whole structure is known as the epigenome.
The whole lineup of histones with
Right.
methylation.
So the histones and the methyl tags, that tongue twister, those two things together are the epigenome.
Got it.
Yeah.
Wild.
Click back in my slide here. Okay, so one more example, just to drive this point home. A mouse study. What they did was they took a male mouse and they trained this mouse to associate a specific odor with a foot shop. Not a very nice experiment, I know, but it works. This mouse learns this very, very quickly, and it's a lasting memory. Every time that odor is presented, the mouse will freeze because it knows the foot shop is coming. When you then breed this mouse, both the next generation and the following generation are inherently afraid of that odor. So what ends up happening is there's a specific gene for an olfactory receptor that's specific to this odor, and the methyl tags have been removed from the experiences of the first generation. So that gene is now expressed in subsequent generations, and those mice inherently fear this odor. Okay? So now we bring up the subject of heritability. What is passed down from generation to generation? The heritability of a disorder refers to the proportion of observed differences that's attributable to genetics. So for something to be 100% heritable, all of the differences have to be genetic. So for something like eye color or connector. If something is 0% heritable, all the differences must be due to environmental factors, like catching a cold or breaking your leg. Those things have nothing to do with genetics. Most disorders fall somewhere in between. Most disorders are a certain percentage genetic and a certain percentage environmental. So how do we determine heritability? There's a couple ways we're going to talk about. The first is family history. And I'm going to use the KE family as an example. Has anyone heard of this family? Okay. It's a pretty famous family in language research. And so this is, again, their genetic family history. And just as a reminder, our squares are males, circles are females, black is affected, white is unaffected, and the diagonal line just indicates that the individual is deceased. So this is a family that for 3 generations has very severe speech and language disorders. And we're going to talk about them in a few more slides, but I just want to show that we can look at their family history across generations. You can look at how many individuals are affected in each generation. Does it show up in every generation? Does it skip generations? And we can determine a heritability probability based on family history. The other way is through twin studies. These are pretty common in research. And what we do is we look for monozygotic twins, so identical twins, and dizygotic twins, or fraternal twins. And you compare how many twins have the same disorder versus how many twins, one twin has the disorder and the other one doesn't. Does that make sense? So in any of these measures, if the identical twins are more alike than the fraternal twins, it suggests a genetic component to the disorder. So for example, in a dyslexia study, fraternal twins with different DNA had 38% concordance for dyslexia. So in twins, 38% of those twins both twins have the disorder. Does that make sense? Okay. And in identical twins, you have a 68% concordance for dyslexia among identical twins. So it suggests that dyslexia does have a strong genetic component to it. And this is just a summary of some twin studies that have been done. I won't go through every single point, but you can see on the top panel here that some disorders like reading disability or autism, you have a very strong genetic component in the identical, as well as in the fraternal. Whereas disorders like hypertension or diabetes, there is a genetic component here, but it's not as strong. So it suggests that there's more environmental factors in those disorders compared to disorders like reading disability or autism. So what does heritability not tell us? It doesn't tell us which specific genes are involved, just that there is a genetic component. It also doesn't tell us how an individual person would be affected, only the average involvement of genes. So if reading is 50% heritable, that doesn't mean that 50% of all cases are genetic. It also doesn't mean that every case of reading disorder is 50% attributable to genetics. It's more of a probability or a running average. It gives us an indication that dyslexia has a genetic component, but it doesn't tell us person to person what's the cause of their reading disability. So it's a piece of the puzzle. So how do genes and your environment contribute to causing these kinds of disorders? So this is a twin study on the genetic influence of reading. They were looking at kids with reading disabilities and the variety of factors that may have caused those issues. So just to kind of orient you to the graph, the white bar, I don't know how easy the colors are to see from back there, but the white bar, the one that's circled, is genetic contribution. The light gray bar is the shared environment of the twins, and the dark gray bar is the non-shared environment between the twins. And what they found was that there's a significant correlation between parental education and genetic influence on reading. So the kids that have high parental education are the kids that have a lower environmental influence and a higher genetic influence.
Say again?
The kids that have parents with a high level of education are the kids that had a high genetic influence on their reading disability, and the kids that had a low influence from their environment. So in those kids, the genetics play more of a role than their environment does.
So environmental effect must be a negative effect. So the high education of the parents must have removed the negative effect?
Well, so the education of the parents would have changed the methyl tags. So it would have created markers on the epigenome or taken them away that then was passed down to the children. Yeah?
So I'm totally amateur in this area, but could I just ask you, the way intuitively I thought about that, which could be completely wrong, okay, is that if you're in a high support environment, which is correlated in a statistical sense, you know, then you're having all the environmental support, and what's left over is the genetic
Yes.
risk.
That's exactly what it
And
is.
that can't be solved by the environment. That's in a sense, to a certain extent, determined by the genes that are present.
Yeah, that's exactly
And
right.
conversely, if you're in a more variable environment where there's lower support, in a broad statistical sense, Now the environment plays a bigger role because it's not all homogenously terrific. Some kids are going to have, a terrific home environment, even if it's a low household without much education. Other kids are going to have a very tough environment. And now the environment starts to become a bigger player. Is that an okay way to think about it?
That's exactly the right way to think
Okay.
about it.
Is there an interplay with poverty in this and socioeconomic status?
Generally, SES in reading disorders, and correct me if I'm wrong, Joanna, is typically education level. I'm sure poverty does have a role in it, but I'm not familiar with those studies. Joanna, do you?
I am just going to confirm that, and I also will add that when people measure socioeconomic status, or SES, there are a couple of different indicators or tools that they use to do that. Some of them are just based on parent education. Others integrate income directly when people are comfortable reporting that. Others use the job as a proxy for how much money they make. So if you report what your profession is, there are databases that researchers can look up what amount of money typically is linked to that profession, and therefore how much financial resource is available to the family. So often people say SES, they don't differentiate between education and money unless the studies specifically do one or the other. But basically these studies speak to if you're in an environment then that can be optimized because you have financial resources to optimize environment, then you can't, there's nothing left over to optimize environment-wise. Whatever you can explain will be genetics. And in a context where you cannot optimize the environment has a lot to do with it. So that's the third way to say already what everyone else said, yeah.
And again, to that same point, your genetic influences can be stronger in risk environments as well as in enriched environments. Because again, we start talking again about the epigenome and your methyl tags and you being responsive to your environment. If you're in a risk environment, your methyl tags are going to change. Same way as if you're in a very enriched environment, your methyl tags will change and your genetic expression will likely change as well. Okay, but which gene influences reading? Why we're all here, right? Reading and genetics. So we're going to come back to the KE family. I mentioned this is a family, very famous family in language research. It's a 3-generation family. They have severe speech and language disorders. They have grammar issues, language issues, articulation issues. The predominant issue in this family was the articulation problem, so oral language speaking. And the affected family members had a lower IQ. So researchers swooped in and they said, this is great, it's a 3-generation family, they all have the same disorder. Maybe there's a gene here. We can find a gene that's common in the affected individuals, and this will be the language gene. So we come up with FOXP2, which has been known as the language gene. Has everyone heard of FOXP2? Has anybody heard of FOXP2? Okay. So I put this in quotes. Why did I put this in quotes? Hey, it's not the only gene, okay? This was huge, saying FOXP2, it's the language gene, it's the one responsible for language. Of course it's not the one responsible for language. It'd be great if it was. FOXP2 is a very large gene. It's 2,500 base pairs, and one base pair is responsible for the mutation. That's it. Okay? This family has one little SNP that causes these dramatic language issues. And what FOXP2 does is it creates a transcription factor that regulates other genes. This is one of the clusters of genes that FOXP2 regulates. Just one of the clusters of genes. So FOXP2, it's a great step forward. It's an important piece of the puzzle, but it's not the whole puzzle. So we do, though, now have the starting point to delve into the genetics of reading impairment. FOXP2 is certainly involved. What are the other genes that FOXP2 regulates, and do any of those genes have anything to do with reading and language impairment? So dyslexia. Why, again, we're all here to talk about dyslexia. You all probably know these things, but I'm just going to briefly talk about what dyslexia is. It's affecting 5 to 10% of the population. Of course, it depends on who you ask and what population you're studying. These kids have normal nonverbal intelligence, but their reading scores are 1 standard deviation below their peers. And we won't go into the phonological deficits and all that because you've been talking about that for the last 2-3 days. There are several genes of interest that have come up in genome-wide association studies. These are a few of them. I'm not going to talk about every single one. One of the key points, again, to mention is that not all people with dyslexia have a variant in any of these genes. or even one of these genes, or all of these genes. But there's a pattern here, and that pattern is neural migration. At least four of those candidate genes are involved in neural migration. So, quick neurobiology development lesson. When the fetus is developing in the womb, the brain grows from the inside out. So you have this area in the center of the head called the ventricular zone, and this is where neurons are born. And once a neuron is born, it migrates out to its eventual position. So these vertical neurons here, these are called radial glia. And basically what this is a tightrope. So a neuron's born, it attaches to a radial glia, it follows that glia until a chemical marker tells it it's time to stop. It detaches and then basically stays there for the rest of its life. And again, the brain grows from the inside out. So the first layer is the most internal layer, and the sixth layer is the outermost layer. So the brain goes from the inside out. At least 4 of the genes involved in dyslexia help monitor this process and make sure that it happens the way it's supposed to happen. So, for example, this is a slice of brain. The dark spots are neurons. They've been stained so you can see them easily. And in normal cortex, neurons are pretty evenly spaced. In postmortem studies of individuals with dyslexia, way back in the day, when they were first looking for a biological basis for this disorder, they started noticing that individuals with dyslexia had clusters of neurons where they didn't belong. So instead of a normally distributed set of neurons, they had these clusters of neurons, and those are called ectopia. And that was very common in individuals with dyslexia. Once we started analyzing the genes and starting to tie which genes are involved in these processes, we were able to replicate this. I say we, the field, was able to replicate this in rats. So this is a rat brain where one of the candidate dyslexia genes was suppressed, and you end up with the exact same clusters of neurons where they don't belong. So we've mimicked the physical abnormalities in the brains of individuals with dyslexia. So I said that there were 4 candidate genes involved in migration. I'm only going to talk about 3 of them. The reason is that the 4th was identified in a very specific population and has not been replicated. So I'm going to kind of put that one to the side for now. DYX1C1, this is dyslexia susceptibility candidate gene 1. It's located on chromosome 15. It was discovered in a Finnish family, but it hasn't been consistently replicated, so we're not going to talk about that one a ton. There's a lot of rodent work being done with this gene, but not a ton of human work supporting it. KIA0319 and DCDC2 are right next to each other on chromosome 6, so they get linked a lot when we start talking about genetics of dyslexia. KIA0319 was named after the group that discovered it, which is nice for them, but a little boring for us. We don't have to find that acronym for that one. DCDC2 is double cortin domain containing 2. Not super important in the grand scheme of things, but that's what it's called in case you were wondering. These genes are the more commonly associated with dyslexia, but there's not yet a consensus about which SNPs are the most important. So again, remember I said how we have 10 million SNPs in our genome. These genes typically have multiple SNPs in any one individual. We've not yet figured out which one is the pathogenic one. So what is the link between neural migration and reading? It's great that we have this physical abnormality in individuals with dyslexia. We're starting to figure out what the genes are, but how does a gene involved in neural migration cause a reading impairment? Another question is location important? Remember, these genes are in every cell. So if this candidate dyslexia gene was mutated in, let's say, somatosensory cortex or your motor cortex, would that also affect reading? Or does it have to be in the language area? Okay, so I promise we are working our way there. How does disrupting a gene affect reading? We're going to use KIA0319 as an example. It's the most commonly studied gene in dyslexia, and it's also the one that I studied, so it seems like a good one. There was a study that came out in 2012. It's the first study, and to my knowledge, the only study linking a SNP in KIA0319 to a specific human brain pattern or behavioral issue. It's the only one that's ever been done, and I don't know why it hasn't been replicated. We remember what SNPs are? I've been saying it every slide, so hopefully.
Switch nucleotide pairs.
Anybody else? What is SNP?
No, sorry.
You should know.
Variation.
It's a variation of the gene. You don't have to remember. What it stands for is single nucleotide polymorphism. Doesn't matter what it stands for, really, for your purposes. All you need to know is that it's a switch, right? Okay.
Tracy, you might have said this before,
Yeah.
but it accounts for a huge amount of variation amongst us, right?
Yes.
The-
Yeah. There's 10 million SNPs in every individual, and 1% of our genome is different among individuals. So it's a small percentage, but it accounts for a huge amount of variation.
You can't throw one out, sorry, I'm messing you up. I can't resist the number of genes, the percentage of genes that are expressed in the brain. You already talked about it.
I did not, actually.
Yeah.
And I don't know the number off the top of my head.
I looked it up here to double check.
Yeah,
I
please.
the right number.
Please.
80% of genes are expressed in the brain, which is by far, as far as people can know, among all the organs we have, lung, heart, everything else, but the organ that has by far the most number of active genes operating in it. 80% of the genes that we have are operating in our brain, which is unbelievable.
But it also makes sense too. Our brain is certainly the most complicated organ that we have. And one interesting thing, I'm going to piggyback off of that a little bit, is that our genes also serve different functions throughout our lifetime. So the 80% of genes that may be functioning when you're an infant and developing may now be serving different functions now that you're an adult. I'm not going to talk about that a ton, but it's just an interesting little tidbit that I like. Okay, so the Pinnell paper, 2012. A very specific SNP in T0319 is linked to asymmetrical language processing in the brain. So you put people in a scanner, you present speech to them, and individuals that have this SNP have a weaker activation in an fMRI scanner in the left hemisphere of the brain compared to individuals that don't have this same SNP. Not only is it related to neural processing, but it's also linked to behavior. So the individuals that have this SNP also scored lower on a reading task compared to individuals that did not have this SNP. So what is this gene doing to the brain? Specifically, on a neuron-to-neuron basis, what is this gene doing to cause abnormal reading? Reading, as we've talked about, is the process of associating written words to their auditory representations. So taking an auditory phoneme and mapping it to the letter, and I won't belabor that point because I know you've been hearing nothing about that point. So, here is a diagram of the human brain. Our primary auditory cortex is tucked right in there. It's known as Heschel's gyrus. And the primary auditory cortex, now this is the first stop that a sound makes in your cortex. This is not a language center, this is just sound. So a tone is processed here the same way as speech. Here it's just a sound. In primary auditory cortex, on a neuron-to-neuron basis, each neuron likes to fire to a specific frequency the best. We call that the characteristic frequency. Sorry, I skipped something. The pattern of brain activity in primary auditory cortex can tell us which sounds are distinct and which sounds are more similar based on looking at the patterns. So I was mentioning the characteristic frequency of cells. So I know this is kind of hard to see in the back, but there's a circle here around what's called A1, or primary auditory cortex. And the colors are the frequencies of those sites. So for example, the neurons over here on this end really like high frequencies, and the neurons on this side really like low frequencies. And so they fire almost preferentially to specific frequencies. Now this is primary auditory cortex again, this is not a speech center. Is everyone following me so far on this? Okay. Okay. So, if you play speech sounds, consonants evoke unique patterns of activity in primary auditory cortex, which makes sense. You'd want a very unique representation of a sound so that you could tell what that sound was. Does that make sense?
Yeah.
These are recordings from a rat brain. And again, each of these little tessellations is a recording site. And again, you've got the low frequency neurons that are grouped together. The high frequency neurons are grouped together, and then there's this gradient in between. So they're organized very specifically in primary auditory cortex. And if you play consonant sounds, the pattern of response is very unique based on each of the sounds. So I'm going to play this, and hopefully it'll go. Oop, maybe I have to actually click. Oh, it's up there, that's why. Huh. Okay. Okay, and this will cycle through, this will repeat so you can see it over and over again. What you'll notice, let's look at bad and dad first. So the sound bad, you'll see that the low frequency neurons down here fire first, and then the higher frequency neurons fire a little bit later. Whereas in the sound dad, the opposite pattern happens. These high frequency neurons fire first, and then the low ones fire a little bit later. So it's an opposite pattern of activation over time. So B and D, for example, evoke opposite patterns of activity. So these two sounds are very easy to tell apart. But if we look at sounds that are more similar in the English language, like R and L over here, or M and N, their patterns are much more similar compared to some of the stock consonants. These sounds are more difficult to tell apart behaviorally because they evoke similar patterns in our brain. Everyone seeing that a little bit? Okay. And we can measure how unique the pattern of activity is in the brain, and we can correlate that to behavior. So pairs of sounds that have unique patterns in the brain are easy to tell apart, and patterns that evoke similar sounds that evoke similar patterns are more difficult to tell apart. Okay? Obviously this is in the normal brain, and the brain responds the same way every time. So we have the stable representation of a sound. If I say the word dad to you 20 times, your brain's going to respond basically the same way every time. You're going to know, I heard dad, that's what it was. But what if the brain didn't respond the same way every time? The auditory representation of a sound is inconsistent. It's going to make it really difficult for you to tell what you just heard. There's a study that came out fairly recently in Nina Krause's lab in Northwestern, and what they did was measure the auditory brainstem responses in kids with dyslexia versus typically developing children with assisted-speech sounds. And what you'll see is each row is a different trial, and the responses are different. This is the exact same sound they presented every time, but the response across trials is different in kids with dyslexia compared to typically developing individuals. Now in humans, we don't get to manipulate their genetics. I have never met, and probably will never meet, a parent willing to come into the lab and sign a consent form for me to knock out one of their kid's genes and just see what happens. So we can do all the association studies we want. We never really know in humans what each gene is doing. So does KIA 0319 cause this or not? We won't know by studying humans. So we go to, can a SNP and KIA 0319 causes? So we go to rat models. And this was the subject of my doctoral work, so I'm going to talk to you a little bit about that. We can use rat models to figure out what's happening in the brain. And we demonstrated a direct link between the gene KIA 0319 and unstable representation of sound. So we basically found that this gene causes the variability that I just showed you. And I'm going to walk you through that in a second. The first thing I want to talk about when I talk about rat models is that you'll hear people use two different terms, knock out and knock down. These are two different things, and it's very important that you understand the difference between the two. A genetic knockout means the gene is gone. It's either been physically removed from the genome or it's been silenced. There is no protein coming from that gene. That gene does not basically exist. A knockdown is a suppression. So we're not removing the gene, we're just kind of dampening it, so there's less protein in that animal compared to a normal animal whose genes have not been manipulated. Yeah?
I don't know if this is too long. How do you do that?
Yeah.
In all the cells of your body, I don't think.
Yeah. So the knockout is done by a very long process, and we didn't use a knockout, so we can talk about it after if we want to talk about knockouts. The knockdown is the model that we used. And first I'll tell you why we picked this model instead of the knockout. In humans that have SNPs in KIA 0319, they actually have reduced protein in their brain. It's not that there is no protein, it's that they have some, they just don't have as much as everybody else. So that's why we picked a knockdown model versus a knockout. And the way we actually create the knockout is there is a scrambled bit of RNA. So this is maybe a little bit technical, but you basically inject that little bit of RNA into the brain of an embryo, And what that does is it attaches to the structure of the gene we're trying to suppress, so KIA0319, it basically attaches to it. And in the neurons that have this sequence, that have this kind of dampener, that gene does not get translated from that gene. But it doesn't get taken up into all cells, you're only affecting a portion of cells, which is why it's a knockdown instead of a knockout. So in the cells that take up this plasmid, those cells will not produce this protein. But in cells that don't take up the plasmid, they function as normal. So
Right, yeah, so there'll always be a portion of those cells that won't do that.
yes. Does that make sense?
Yes.
I try not to get too
The
technical
key
about it.
is it has to be an embryo.
It's an embryo.
That's, yeah.
Yep. The surgery is very complicated. Okay, but so what we did was we knocked down KIA0319 in rat embryos. We let them grow up, and then we recorded directly from auditory cortex. So again, the beauty of rat models is we can actually put an electrode into the brain and see what every neuron is doing. Again, no parent is going to let us stick electrodes into their kids. So rats are great for a lot of things. So what we did was we played a variety of consonant sounds. We played the sound D and the sound B. So remember, those are two sounds that are easy to tell apart. We played each sound 20 times, and we recorded the responses. These are the responses in a normal rat, a normal auditory cortex. So what you'll see is each row is a different trial. So vertically, there are 20 trials of D and 20 trials of B. Each of
Same rat?
Yeah, this is the same recording site. This is one electrode.
One rat.
Yeah, one rat, one electrode. The dots are responses from the brain, so action potentials from the neurons, and time is on the x-axis here. So what you'll see in this normal animal is that every time we played the sound D, this recording site responded very quickly and very consistently. It always responds within the first 5 to 10 milliseconds after we play the sound. When we play the sound B, there's a little bit more variability, but generally it responds very consistently over time. So you could pretty much tell by looking at these two which was which, pretty reliably.
So does the top line show that it's the brain's responding less fast to that sound?
To B? Yeah, so you remember when we were looking at the video how to the sound B you have the neurons that like low frequencies fire first, and then the neurons that like high frequencies fire later? This is a high frequency neuron. So what's happening is it fires very quickly when it hears a D, and it waits a little while and then fires when it hears a B. So that way you can distinguish between the two.
So the petition is that the next one, when you do
The knockdown?
the knockdown one, is gonna look very similar, they're gonna be, they can't just
No, it's gonna be messed up. Yeah.
Okay? So you're exactly right. Not only is the variability higher, but there's a delay even to D.
Yeah.
they're not even doing
It's not even picking it up right away, it's waiting. So if I were to cover up B and D, would you be able to tell the difference? No. They're very similar.
Wait a minute, why not? The D's are below the line and the B's are above the line, right?
Okay, if I scrambled them. If I scrambled them up, and I presented, remember each row is a different trial? So there's 20 different repeats of the sound here, and 20 different repeats of this sound. If I took away the marker, if I didn't tell you whether it was a B or a D, and I shuffled them up and just showed you one, would you be able to tell what it was?
Oh, I get
You see what I'm saying?
Yeah,
If you were going to use this one, if I showed you a site that fires very quickly, you'd say it was a D.
So you're showing us by time.
The
That
Yes.
line down the
D
middle
in the
doesn't do
bottom
anything.
could be.
This
a
vertical
D
one
from
is
the
just,
top.
or this horizontal line is just separating B and D. Sorry.
Oh, so you could overlay these things. So B and D are differentiated at the top by time response.
Yes. Yes,
And by the bottom, they're just not differentiated.
that's correct.
But it also looks like
That's
in
my
both
kids.
there's more consistency in the D than there is in the B.
In general, there is because D is
Easier.
it's an unvoiced stop consonant, so it has more intensity at the beginning. So your brain responds when sounds come on in general. So yeah, I mean, you do see a little bit more consistency in the D than in the B. But the delay, this latency here, is going to mess with you no matter what.
Yeah.
Jennifer.
Yep.
Okay.
So we see in our students.
Exactly. Now, this neural issue does translate to behavioral impairment, even in our rats. There we go. Okay. Now, of course, every time I say that, I get the exact same reaction that I just got from you guys. Rats doing speech discrimination tasks. We're not teaching them to read. We're not teaching them to speak. These are sounds to them. And rats are actually very good at telling English speech sounds apart. They're very good at it. They can do consonant discrimination. So you train them to press a lever to the sound D and ignore everything else. They'll press a lever to D and ignore everything else. They can do vowel discrimination. They can do the task in quiet. They can do it in background noise. They can do it in cochlear implant simulations. They can do it in a variety of speeds. And their thresholds are the same as humans. So again, their thresholds at these tasks fall apart at the same place humans do. So their speed threshold is the same as ours, their background noise tolerance is the same as ours. So we can use them as a model for just auditory processing. How's the speech sound coded in the brain, and how does the brain use that information to tell sounds apart? So this is a rat who was trained to press the lever to the sound dad and ignore everything else. So we always have a speaker outside the cage because rats will chew on everything. You have a pellet dish, and then her lever is down here. Dad. So if she presses a lever, she gets a little sugar pellet reward for getting it right. Sad. And she pushed it when she wasn't supposed to, so she gets a little timeout and say, no, that was the wrong sound, don't do that again. The light goes out and the program pauses. So now she's mad and she's not looking at us. Sad. So now she says, you're not going to trick me twice. Not pressing it again. Now the rat hearing range is higher than ours. So we shift our sounds up 2 octaves so that they can hear it. And that's what that shifted voice is. Okay. They're very fast. They're very, very fast.
Yeah.
Okay.
What happens to those rates right now?
The lights go off and we pause the program.
So you just make sure it's in the dark and not.
Yeah, pretty much. And actually, it's funny because rats are nocturnal. So we always used to debate about whether or not they were actually getting the message. We had some rats who would just press the lever constantly so that they could just be in the dark. And then the lights would come back on and they're in a corner sleeping. So come on. Yeah. So some rats manipulate the system a little more than others. They're smarter than people give them credit for. So we trained both control rats, which is the thin line, and then the knockdown rats in the thick line to press the lever to the sound dad and ignore sad, bad, gad, and tad. So it's a constant discrimination task. And this is the full speech one. What you'll see is that there is a little bit of a learning curve. The knockdowns don't learn it quite as quickly. They need about twice as much time to reach the same level of performance as the normal animals do. So when we first saw this result, we thought, well, there's one of 2 things happening. Either they're just bad at speech, or they just need extra practice. So we trained them on a task we know is difficult for kids with dyslexia, which is speech and background noise. And this is a hard task for the animals, but the control animals do eventually get better, whereas the knockdown animals do not improve, no matter how long you give them to practice. Now, all of these sounds up till this point are recorded by a human. They're not computer generated. So there are little subtle differences in the pitch of the sound and the duration of the sound. And rats are smarter, again, than you give them credit for. They can pick up on those subtle little cues. And we know that kids with dyslexia can kind of pick up on those little cues to kind of help them through their day. So we wanted to test that. And what we did was we presented only the consonant sound, only the initial consonant, the first 40 milliseconds of the sound. So there's no more duration differences, there's no more pitch differences, and you see a persistent impairment over the course of training. The knockdown animals cannot figure out that phoneme discrimination task. Now, for the sake of time, I'm not going to go into the follow-up studies. I'll tease you with them, and then if you want to talk to me about them after, I'm more than happy to do that. We did find that 4 months of behavioral training fixed the neural variability impairment, so the brains become more consistent after training. And the order of training matters. So if you train them on the short phoneme task first, they do not have speech and noise impairments. So that's my little teaser. Happy to talk about it after if you'd like to. So human imaging techniques, though, are not at this level of detail. We're making progress. I'm actually working on a study to test these results in humans, and that'll be starting this fall. So look for those results, hopefully in the coming years. Okay, so some take-home points. I know we talked about a lot over the last hour. Human disorders are complex. That's like the #1 takeaway from this whole thing. In most cases, disorders are caused by multiple genes, and disorders can be caused by not only inherited gene variants or SNPs, but also by inherited methyl tags or acquired methyl tags throughout the course of your life. These genetic factors can change the way the brain processes objects and sounds, and a subtle difference in neural processing can have profound influences on cognitive tasks such as reading. So I want to do a quick discussion. I presented these learning goals at the beginning of my talk with some questions here. I'm going to take about 5 minutes, turn to the person next to you and just kind of talk about these things. We'll come back, kind of recap these questions, and then we'll close.
Can you still do that for us? I can't get into class, and I was wondering if I have to do that.
Well, exactly, and if we're looking at the nature versus nurture, right?
Okay, but when we went to it, there was nothing in it. A couple of us tried to get, she sent us a link to go to. It's nothing, yeah, unless I'm doing something wrong. Okay, good. Perfect.
Mind-blowing stuff.
All right, so first question, what does heritability mean? Who wants to tell us what heritability means? What does heritability mean? Yes.
The phenotype that seems to be inherited can be attributed to genetics.
Right. So how much of a difference is caused by genetics? That's your heritability. How are dyslexia and heritability related? Who wants to volunteer for this one? I'm going to pick somebody if you don't want to. You're not going to like it.
Well, there are certain genes that can show a disposition towards dyslexia.
Right. And how do we find that out? Remember the two methods we talked about? How do you determine?
Family study.
And
Twin study.
very good. Very good. Are genes deterministic?
No.
Hey, that was kind of a trick question. Right. So your genetic structure is passed down. That you can't change. Your epigenetics can change. So yes and no. It's a little bit of both.
Yes, no.
Okay. How can epigenetics cause human disorders? Magic. No, it's not magic. Who wants to take a stab?
When they're tagged and untagged, they can be read and unread and determined upon their location, right?
Right. Right. So if you had a gene, for example, let's say the FMR1 gene for Fragile X, if you somehow got a tag on that gene and the epigenome tightened up and that gene wasn't able to be read, you would have Fragile X. That's another way that you could have gotten it. How can a gene influence behavior? We spent the second half of the hour talking about this. So many ways. Pick one.
SNPs.
What's that? SNPs, but how? How can a gene change the brain and then change behavior? What's the link? Neuromigration. Yep. Yep. And the neural variability that we saw.
And the
Right? If you have a gene system.
synthesis, the fragile synthesis.
Absolutely. Okay. So I want to close by just encouraging you all to keep your eye on the research. We're learning more every day, certainly, and we can make all the discoveries we want, but it's really up to you to use that knowledge and improve the lives of kids with reading and language impairments. So thanks for bearing with me through that dense subject, and I'd be happy to answer any questions that you have. I know we ran a little over. I apologize for going over.
So when did epigenetics and mental tags, when did that kind of first get discovered? 20 years out of biology from high
Yes.
school, we didn't know about any of that.
That's actually, I honestly don't know the year. I can certainly look it up. I'd be happy to
In the
look it
last
up
40
and
years.
get back to you. It's only recently become a buzzword in the field. And even within the genetics world, there's a lot of people that still don't really know that much about it. I've only recently understood what it is, to be honest with you. It's still a very new piece of the puzzle, but we were just talking about the nature versus nurture idea, which has been obviously the root of so much of our educational background. And the idea that now, because of this whole epigenetics new field, they're more interlinked than we thought, right? You have your genetic code that's passed down, that's your nature, and your nurture actually affects your genes.
Yeah.
So the two are very linked, and I think we're going to see a lot more of the epigenetic stuff in the coming years, trying to explain away what is the environment actually doing to our genes, how is it changing us.
Does that
And the
begin
other
to explain
question you had-
evolution?
I'm sorry, say it again?
Does it begin to explain evolution?
Yes and no. I mean, methyl tags certainly do get passed down from generation to generation, and I guess to a certain extent, if you had a set of methyl tags that were
Advantageous.
right, if they were advantageous, they'd certainly be passed on. In the sense of, for example, the mouse example, where they trained the mouse to be afraid of the odor, to associate the odor with the tone, there may be a generation where that odor is no longer a bad thing, where maybe that odor becomes indicative of food, for example. An animal that's afraid of that odor is going to starve and die, right? So, I mean, there are certainly ways that epigenetics can be passed down and part of evolution, absolutely. Sorry, one more.
Well, our other wondering was, is there, if those mental tags get passed down, is there, like, say, a stress-induced one gets passed down generationally? Can you undo that through cognitive approaches? Like, could you kind of teach mindfulness and anti-stress and start to see some of those mental tags?
It's called extinction, excuse me. I'm sure that there are some instances where methyl tags are there, and it would take a lot more work to remove them. But there are studies in these same kinds of paradigms where you train a mouse to be afraid of a tone or an odor, for example. With enough training, you can reverse the association. So, I mean, you can do the training, but I think we get now into the world of things like PTSD and things that can be retrained but are more difficult to retrain.
Yes, I look at your last sentence. It's up to us to use the knowledge to improve the lives of children. Yeah.
I got no pressure, no pressure.
No. Okay, so how can this knowledge help us?
Right.
So the only thing that I'm finding to help me is when the kids ask, why am I dyslexic and my sister isn't?
Right.
And I have a lot of knowledge of the families. I often meet uncles and aunts of these kids, and I can spot the dyslexic ones. And one girl, I blurted out, well, your Uncle Hugh is dyslexic. And she went, how do you know? I don't know that. Oh, her mother wrote me quite the letter. We don't talk about that. And, I mean, we can't even help these children to understand their dyslexia by putting out their heritability because it's the generational secret.
Yeah, well, and part of that is, it's the stigma, and we are still working past that. I have, my mother's uncle grew up with dyslexia. And, you know, back then, of course, the stigma was, well, you're just stupid, you're never gonna learn this, and so, you know, he never had a career, you know, went through life feeling like he was stupid. And to him, this research was life-changing. You know, the idea that it's not my fault, the idea that it's biological. And again, I think we are kind of fighting that stigma of like, oh, we don't talk about this is, you know, not something that we're gonna air, the heritability issue, and I think that's certainly an uphill battle that we're still gonna have to climb. There's a really good book out called The Dyslexia Debate. I don't know if anyone's read it, but the idea of like labels, and is the label helping us, is it hurting us? I know you guys have talked about this a little bit. And I think the heritability issue is along those same lines, that we have to kind of break down the barrier that because it's heritable, it doesn't mean that your uncle's stupid either. It just means that this is something that's biologically who you are, but we understand more about what each gene is doing. So for example, if KIA 0319 is actually causing this neural variability issue, there are ways we can treat this. If we can identify an individual early enough, we can train their brain to be more consistent before they even hit school. And so these studies will hopefully help us with some more targeted therapies, understanding what's going on in the brain and what each gene is doing to cause the disorder. But we certainly have more work to do to make it not stigmatized.
My kids, I refer to the students, would love to hear that it is completely inherited.
Yeah.
What about the other 50%? Where do you think that comes from?
Well, again, I don't know the answer. I'm not going to sit here and make one up for you. I think we kind of go back to that whole nature versus nurture issue. I think that epigenetics are going to play more of a role as we start to understand what's happening and how is it changing throughout the course of someone's lifetime. Is it that a child is born without an inherited dyslexia gene mutation, for example? Maybe they don't have the SNP in KIA 0319, but something in their life has caused the epigenome to tighten around that gene, and now it's no longer being expressed. I think we have a lot of work to do. We really don't know the epigenetic component to this, but it's an exciting new area of research, and I think you're going to see a lot of it in the next few years. One more really quick word. Anyone else? One more question? Anybody? I think John had something.
Okay, it's an ethics question, but let's just do the science fiction for a moment.
Yeah.
Could you imagine a day, and you want such a day, where you could have direct biological treatment of these genes in such a way that shortly after birth, if a child has an unlucky set of genes for learning to read, you could ameliorate that. Would anybody want that?
Well,
I think, would
So-
anybody want that, or would you want that to be an option available to parents for their children? Or is that 1,000 years from now, so you're able to think about it?
well, let's take a quick poll, because I'm interested to hear what other people think. If you had the option to change your child's genes if they had risk genes, how many people would do it? How many think that's a good idea?
Without other risks.
Let's say that both parents have dyslexia,
Right.
and therefore the heritability, even though it's a population level statistic, let's say that all you do is at the population level, there's a heritability of 50%. If
It's
both
about-
parents
it's about
had it,
68.
for the population, 68, if both parents had it for the population level. Now, you're two individuals, and you're about to have a child. And if you find out,
Yeah, so your baby's born, you do a genetic test, and your baby has a SNP in Keo 319.
but like knock it out, so you can't do it, if you have to do it.
If you could fix it, let's do science fiction. If you could fix the SNP, put it back to the way it's supposed to be, make the gene.
Without side effects, without side
Well,
effects.
that's the key though, without side effects. So remember, I mentioned this very, very briefly, that all your genes have multiple functions.
Yeah.
So it's not just a matter of fixing the gene, but it's when do you fix it. So if a baby has been growing in the womb for 9 months with this SNP, what damage has already been done? The neuromigration issue is already done.
Anyone more science fiction for a second?
Yeah.
I'll stop it. Next science fiction is, you don't do a change in the child's genetics, but what you do is you develop a drug that promotes the operation of that protein, and you give that drug while that child gets intensive intervention because it would accelerate the power of the treatment.
Well, that'd be a totally.
Power of the intervention. Is that possible?
So yes and no. The problem with, when we're talking about Keo 319 specifically, is that that protein, it's involved in neuromigration, but it also has a function after. So through adulthood, it's actually, without going into too much detail, it's a protein that is on the membrane of the cell. And what it actually does, we don't know. So it's really difficult to answer that question, to say, do you want to promote the expression of that protein without knowing what that protein does? I think at this stage, if you were going to pair a pharmaceutical with therapy, you'd want, a little bit more detail. These cells, if you suppress the function of this gene, the cell is hyperexcitable. So it'll fire more often than it should. If you could induce a drug that calmed down that firing and made it a little less sensitive, for example, I think that could be effective. But you don't want to just pump in more protein without knowing what it does. So we've got a lot to learn.
Yeah.
But we're making progress. So.
I know we're a little late, later, so that we want to stop today, so hopefully it was very enriching and you feel like there's lots to think